Hi guys, this is Miss P, and I'm here to give you guys a little bit of a tutorial about DNA, RNA, and protein and how all of those things work together in our cells. This week's instructional videos are going to be done in two parts, so this will be the first part, and then there will also be a second part that goes into a little bit of specifics about RNA, specifically about kind of what it is, what it looks like, and what it does in our bodies. So look out for that video too. You'll want to watch both of them before attempting that practice worksheet in task 4. As always, I have some learning objectives here. These are the things that you need to know or need to be able to do. This first video is going to focus on the first learning objective. So basically, can you explain the relationship between DNA, RNA, and protein? That's what you should be focusing on for the purpose of this first video. So our big kind of deal this week is looking at reading DNA. So we've studied DNA, we know about its structure already, we know about how it replicates itself, so how it copies. Now we're going to figure out how our bodies actually read that DNA and how they use it. So this is kind of the cool part about DNA, in my opinion. So basically, DNA is like a recipe. So I want you to think about your favorite food, dessert, or dish. It can be something that you know how to make, something that like your parents make for you, something that you only have at special occasions. For me, this thing is called sour cream chocolate cake. It's my grandma's recipe, and I want to know how to make it. So if you wanted to make your favorite dish, how would you know how to do that? How do our cells know how to make the things that we need? That's kind of where we're going today. So like I said, DNA is like a recipe. So if you wanted to know how to make your favorite dish, but you didn't know how, but you knew somebody that knew how to make it, say your mom or your grandma knows how to make it, you could call them up. You could use your phone. You could call them up. You could say, hello, I wanted to make that chocolate sour cream cake that I think is so delicious. Do you have the recipe for that? They would say, most likely, yeah, sure, let me get that for you. And then they would grab their recipe and they would read it off to you. And you would write it down, maybe on a recipe card, maybe just on a scratched piece of paper. You would write it down in some way. And then you would take those instructions that you wrote down and you would use those instructions to make whatever it is you're making. In the same way, DNA works similarly to that. So we start with our DNA. Think about our DNA as our original recipe. You might not have the instructions right away if you're just learning how to cook this wonderful dish that you're thinking about. So you might have to call someone. They would give you the recipe over the phone, so you would have to write it down. So there's a process happening here between DNA getting to RNA. So this process is called transcription. Transcribing something is basically writing it down. So we're taking our DNA and we're writing it in a new way. And we call that new version of our genetic material RNA. Very similar to DNA. It's a little different. We'll learn more about that in our second video and throughout the time today. So after you have your recipe, you've written it down, you're going to use that recipe that you wrote to make your final product, which in our bodies is called protein. So these proteins are actually the molecules in our body that make us who we are, that make all of those phenotypes that we've been talking about in our genetics unit. So we talked about DNA, right? DNA is our genotype, and it gets expressed in our bodies through proteins. So proteins are actually the thing that cause those phenotypes, that cause those outward characteristics that we can see and that we interact with on a daily basis. And the intermediary between those two things is called RNA. So just like I've been saying, we have this pattern in our bodies. So just like we would make a recipe in real life, our cells follow this similar sort of pattern, a similar sort of recipe to make us who we are. We call that pattern central dogma. So central dogma is basically described as DNA is converted to RNA, is converted to protein. So it describes this two-step process for how our genes become expressed. So our genes are in our DNA, and they are expressed through central dogma, and then our proteins are actually the things that make those traits visible to us. So there's two steps here, basically, right? There's two times where something happens to our genetic material before it becomes a protein. which causes us to be able to see those traits. So that first step is called transcription. So transcription happens in the nucleus of our cells. As eukaryotes, we have a nucleus, and transcription happens there. So during transcription, an enzyme called RNA polymerase binds to DNA, opens up the double helix, and uses that DNA strand to write or transcribe a single strand of mRNA. So RNA polymerase is kind of like DNA polymerase. Remember, we talked about DNA polymerase last week? DNA polymerase was that enzyme that binds to our DNA and adds nucleotides to it to create another copy of DNA. So very similar, RNA polymerase is also adding nucleotides, but it's adding nucleotides specific to RNA. Let's use an example of this to kind of like walk through it a little bit more. So here's our example. We have our DNA strand, our original recipe, and we want to rewrite it so that we can use it to make proteins. So we need to change it from DNA to RNA. In RNA, when our RNA polymerase adds those nucleotides, it's going to add nucleotides specific to RNA. So for DNA, we have nucleotides like A, T, C, and G, right? We've talked about those adenines, thymines, cytosines, and guanines. In RNA, there's another nitrogen base. So there's another kind of nucleotide that we get to use when we're talking about RNA. That nucleotide is called uracil. So uracil in RNA takes the place of any thymine base. So uracil is going to be the base that binds with adenine now. It only binds with adenine in RNA molecules. So you're only going to see U's if you're looking at an RNA molecule. If you're looking at DNA, you're going to see T's. But in RNA, we have uracil instead of thymine. So we're going to see U's instead. So as we transcribe, we're not going to just automatically change like T to U, A to A. We want to make a complementary version of this original DNA strand. So what I mean by complementary strand is I'm looking for the bases that would bind with this base. So the complementary base of a T is an A. So our first letter of our mRNA code should be the letter A. Our second letter in our DNA code is an A. So normally, if we were just making another copy of DNA, we would be able to see a T here for thymine, because thymine and adenine always bond together. But because we're working with RNA right now, we're not going to see a T here. We're going to be using the nitrogen base uracil instead. So we're actually going to see a U bond with this adenine. So our first two letters of our mRNA code are A and then U. I want you to take a couple seconds at this point, maybe a minute, pause the video. I want you to finish transcribing that DNA code into an mRNA. So we're looking for those complementary bases. And remember, we've got uracil instead of thymine, so I shouldn't see any T's in your mRNA. I should see U's instead. Take a couple minutes, pause the video, work that out, and then come back to us. All right, welcome back. We're going to go over our answer for our transcription right now. So what you should have here, let me see, do I have it? I don't have it on there. Okay. So what you should see in your mRNA strand, listen closely. You should see an A, a U, and a G for the first three letters. So the first three letters are A, U, G. The next three letters should be C, A, C. That's C, A, C for the second set. And the third set should be U, G, and A. So U, G, A are the final three for mRNA. So the whole line should read A, U, G, C, A, C, U, G, U, G, A. You'll notice that I've grouped all of these letters into groups of 3. So when we think about DNA, we're going to think about our DNA code words, the words of our DNA, being 3 letters long. So every 3 letters of DNA is called is a word of DNA, and we call those words codons. We're going to talk about those more this week as well. So that was step 1. Step 1, we took our DNA and we transcribed it, so we wrote it down as RNA. In step 2, we can take that RNA and we can translate it into a protein. So what happens during this step, this translation step? So during translation, we're going to see this happening in the cytoplasm. So transcription happened in the nucleus of our cells, but translation happens in the cytoplasm. So after our DNA was transcribed into mRNA, that mRNA traveled through the membrane of the nucleus into the cytoplasm. And once it's in the cytoplasm, it can attach to a structure called a ribosome. So a ribosome is an organelle, and we know that this organelle is the site of protein synthesis. So it's where proteins are made, which makes sense because that's what we're on. That's the step we're on. We're translating our RNA into protein, so we're going to do that in the ribosome. The ribosome is going to read that mRNA strand, and it's going to interpret it, and it's going to signal something called a tRNA to bring in the correct amino acid. So over here, we have a picture that's kind of describing what's happening in translation. So we have our mRNA strand. This is the thing that we transcribed in the nucleus from our DNA code. It exited the nucleus and it attached to a structure called a ribosome, this teal blue colored molecule here. Once it was into the ribosome read that DNA code and it signaled something called a tRNA, which is right here, to bring in an amino acid that matched whatever code the mRNA had on it. So this amino acid, this blue amino acid, stands for the letters A, C, and G. And this strand of amino acids right here is called a protein. So proteins are made up of a long string of amino acids. So you can kind of imagine translation as adding beads to string. You remember when you were in like first grade or kindergarten or preschool and they had you make beaded necklaces or necklaces out of macaroni? This is the same kind of thing happening. We're adding our amino acids to our chain and we're making a protein. So let's do an example of this so we know kind of what this looks like, because I'm going to be asking you to walk through this during that practice worksheet in task 4. So we're going to do an example. So just like we had before, we're going to have our mRNA strand. This is the same mRNA strand that I had you transcribe earlier. So we had our DNA before, we transcribed it into mRNA. Here's that mRNA code we had. Now, I want you to be able to take that mRNA and put it into tRNA, and then also into amino acids. So how do we do that? How do we make this happen, right? So for mRNA, we're going to look for its complementary bases for our tRNA, because our tRNA is going to have, our mRNA is going to be the code that we use to figure out what kind of tRNA we're bringing in, and what kind of amino acid comes in with that tRNA. So we're looking for the complementary bases to mRNA, and that's what we're going to write down for tRNA. Now remember, we're still talking about an RNA molecule here, so we're going to still be using uracil in place of where thymine, or T, would usually be. So when we do this, we'll do this first one together, and then I'll have you do the last 2 sets on your own. But this first one, what is the complementary base for adenine, or A? What's the letter of the base that always bonds with adenine? We know that that's usually thymine if we were in DNA. So if we were going back to DNA, we would use a T here. But we're not. We're in RNA. So we're going to use uracil instead. So for our first letter of our tRNA, it's going to be a U. The second letter is going to be an A, excuse me. So we have a U and an A, and our third letter, what bonds with G always? We know that's cytosine, or C. So our first three letters of tRNA are U, A, C. All right, I want you to take a minute here and pause the video and translate these letters of mRNA into tRNA. Don't forget, uracil is taking the place of any thymine that you would usually use. All right, now that you've finished doing that, we'll go over our tRNA, and then we will talk about how to figure out our amino acid chain. So for our tRNA molecule, we should know the first three already. We did those first three together. We should have a U, A, and C for our tRNA. For our second set of three letters, our second word, if you will, we're going to have a G, a U, and another G. So G, U, G. And for our third word of mRNA, our third codon, we're going to have an A, a C, and a U. So our whole tRNA strand from front to back should read U, A, C, G, U, G, A, C, U. And there it is, our final tRNA strand. Now that we have our tRNA, we should be able to figure out what our amino acid chain is. So remember, our amino acids are those building blocks of protein. So they're the things that are going to actually make up our protein. Just like nucleotides make up DNA and RNA, amino acids are the things that make up proteins. So we have 20 different kinds of amino acids in living things, and they're all included on this chart right here. And they've got some really fun names that are kind of hard to say. Don't worry about how to say them. I will always have you writing them down. I won't have you saying them. You will never have to memorize this chart. This chart will always be provided to you. I will never expect you to memorize these. So when we're looking to translate our final RNA into our proteins and our amino acids, we are going to be using the mRNA strand to find our amino acid bases. Because remember, when we are translating, our ribosome is reading our mRNA to determine which amino acid to bring in. The tRNA has a complementary set of nucleotides, so it can bond to that mRNA, which allows the amino acid to attach. But we're actually reading the mRNA strand when we translate. So we're going to look for these letters to determine what amino acid is the first amino acid. So when I'm looking at this, there are three letters, right? Our chart has areas where there's a first letter, a second letter, and a third letter. So we're going to use those things to help us figure out what amino acid we're bringing in. So our first letter of our first word is an A. So I know that our first letter is an A. So the one I'm looking for is going to be somewhere in this row. My second letter is a U. So I know it's in this row, and I know that because U is in this column for the second letter, this is the column I'm going to be in. So now that I know that, I know my codon, or my amino acid that I'm looking for, is somewhere in this box. And now I can look down my list and find my third letter, which is G. Here are the three letters of my mRNA that I'm looking for. This is the name of the amino acid that it's bringing in. The name of this amino acid is methionine. Usually we just use the first three letters to abbreviate them. So you can call it methionine, you can call it MET. The fun thing about this one is it's the start codon. So this is the codon that's going to be at the front of any translated protein that you ever find. So our first amino acid is methionine, or MET. For our second amino acid, we're going to look at our second codon, our second set of three nucleotides. Again, we're still looking at our mRNA, not our tRNA. We're looking at our mRNA. and we're going to look for C, A, C. So our first letter is a C, so I'm going to go to the column that has a C as its first letter. So we're somewhere in this row. Then I'm going to go to my second letter. My second letter is an A. So I know I'm in this first row for my first letter C. And if I follow along on the top, my second letter is an A. So it's somewhere in this column and this row. So I know that the amino acid that I'm looking for is in this box. So it's either histidine or glutamine. It's all dependent on that third letter, which in our case is a C. So I'm looking for C, A, C, which is right here. These two codons both code for the amino acid histidine. So I could have had a CAU or a CAC to get the amino acid histidine. This is the second amino acid in my chain. So first I had methionine. My second amino acid, coded for by this codon, is called histidine. I want you to try the third one on your own. So look at your third codon, your third set of 3 nucleotides, and I want you to look for what amino acid this codes for. Pay attention to the first letter, then the second letter, then the third letter, and write down what kind of amino acid or what kind of codon we're looking at here. Take a couple minutes, pause the video, and work on that. All right, welcome back. I'm glad you took a little bit of time to work on that. And we're going to go over how you figured out that last amino acid, or that last codon, I should say. So we looked at our first letter, which is a U. Here's the row where the first letter is U. Here's our first letters. U is our first letter. Second letter is G. If we look along the top where our second letters are, G is all the way to the right. So we have a U and a G. So it's going to be something in this box. And it all depends on that third letter. In our case, that third letter is an A. So we're going to follow over here. We've got our U, C, and then here's our A. So UGA, as an mRNA codon, is called the stop codon. So this is what you're going to find at the very end of any protein that's made through translation. And there's a couple different versions of the stop codon. They all do the same job, but there's just multiple ways of saying it. There's a little bit of wiggle room, if you will. Remember, each protein starts with methionine, or AUG, but they all end in a similar way too. So now that you've got that amino acid chain written down, you should have methionine, or start codon, then you should have histidine, and then our stop codon. Let's see if we got that right. Yes, we did. Notice we're abbreviating those amino acid names with the first 3 letters. That's totally fine. You can write out the full name or you can write out the shortened version. Both are fine as long as I can tell which amino acid you're talking about. So these 2 examples, our step 1 example about transcription and our step 2 example about translation, are what I'm going to be asking you to do on that task 4 worksheet. So if you get stuck with that worksheet and you don't know what to do, come back to this video and rewatch it. Go through those examples again. Listen closely about what exactly we're doing and when we're doing it. All right, congratulations. This is the end of the first video. You've made it. Whew! That is everything that I expect you or would need you to know about getting DNA to RNA to protein.